branded strategy is there. By using the gaming of patent, they prolong, they block first to file generics launching. And this is the perfect example of using the gaming of patent, FTC Cephalon. Cephalon company, this is the branded, Cephalon company, how is that working? Oh, yes. Cephalon company in 1998, this company had got the approval of the Provogil. This is the branded wakefulness. Modafinil, this is the API, generic. And after 4 years, 4 generic companies, they have filed for that. But because of this settlement, they have prolonged this up to 2012. So this is the strategy of brand, how to prolong the launching of this F2F, first to file. Average time, it keeps coming to 9 to 11 years, whereas a para 4, that can go up after 4 years. Last 20 years trend, this is a trend of market exclusivity period. In 1995, this it was difference. Exclusive period was 13.5. Now it has come to 12.9. Whereas this, if molecule is more than 100 million cell, that comes to 12.6. This trends for para 4 challenge for last 20 years. In 95, only 9% molecules was experiencing this para 4 challenge from the generic, whereas these days, in 2012, it has come to 81%. This is steadily had increased the competition, and if you see that's time, brand to para 4 challenge difference time is 18.7 years was there. Now it has come down to 6.9 years. This is the stiff competition between the generic and brand is there. QBD challenge and way forward. I mean, how to implement the QBD challenge is divided, comprises of 3 steps. Steps 1, that is steps 1, this focuses on process understanding, and steps 2, regulatory challenge, which is how to implement the trust of the regulatory harmonization, and step 3 is the regulatory flexibility. Post-approval benefits, full benefits, company can experience with this. The key features of process understanding is that how to develop the robust formulation. In order to understand what is the robustness, we need to know the total variability of the product. A product is made of 3 different components: active, API, excipients, and process. Earlier approach, in order to get consistency, input parameter for API plus excipients, that is to make it very tightly. This is called a reproducibility in order to get consistency. But these days, in order to considering this variability on the part of this active as well as excipients, process endpoints is to be adjusted. This is called robustness. Industry impediments to QBD adoptions. Why industry is resisting for the QBD adoptions imposed by regulatory affairs, regulators? Because QBD, quality by design, doesn't address these 3 particular part in the initial stage: COGS, cost of the goods sold, sustainability, scalability, and there's no assurance from the regulator for if it is followed and it's getting failed in clinical trials, regulatory review, or commercial stage. Generic company status is like that; they are confused state. So, what is the option to the generic company? What to do with this? what the study is supposed to be the generic, the goal which is supposed to be that to develop a mix equipment manufacturing strategy that maximizes quality, scalability, sustainability, which QBD doesn't address, with an eye towards cost effectiveness and commercial viability. And it is possible by this is the strategy for the thinking beyond QBD. And thinking beyond QBD, it is possible by rapid process understanding, exceeding the requirement of the QBD, and getting off the critical path onto the right path, which is a precise and sensitive in nature. And these are possible by trapping, linking, capitalizing the opportunity which is available in the environment. What are the generic opportunities that there? We will discuss now. Single cycle development. Currently, the step, currently development activity is like that. I mean, R&D lab scale, pilot scale, and subsequently after these activities goes to the commercial scales. And this follows the 10x formula. From this to this 10x, batch size supposed to be there. From this to commercial scale, that's supposed to be 10x. And this is the source of variability. And by eliminating scaling up, I mean the same equipment, if it is used, same equipment of pilot plant is used for the commercial scale, variability can be minimized, can be eliminated. Using the same equipment for R&D to scale up the commercial production will cut cost, time, improve, and variability as well, it will be reduced. And moreover, in the same run, the engineering runs as well as this validation run can be continued. This exhibition batch, which is a clinical batches, at present this is being carried out by in the pilot scale, that will be treated in the commercial facility as well. Single-use technology approaches. It is based on the disposable technology. It provides the operation flexibility, no CIP, no SIP, and changeover time is also very less. Tank to needle concept directly, and time to first manufacturing batch times, if you consider it comes to 4 to 13 months. Reason is that there is no need for the cleaning validations for disposable equipments, disposable parts, and moreover, procurement equipment is also comparatively it is less. And cost-wise, manufacturing cost is also comparatively very, very less. Reason is that there is no, I mean, API saving is over there in this step, which outlays this cost which is required for the every batches over here. Continuous processing. This is the another opportunity, generic opportunity design, process design stage, and game of the PAT, process analytical techniques, and of the QBD, a changing processing the paradigm. Currently different activity, all activity having the different equipment, and their batch size is different. And each step is scaling up as per the batch size of the concerned equipment, which is the source of the variability. Whereas if we go continuous manufacturing, here batch size that's supposed to be always constant. There is no source of variability because batch size fluctuation is not there. RTRT is another strategy, real-time release testing. This is the testing paradigm. Offline testing in the QC and product testing, whereas the online testing, by adopting this, the real-time QA, quality assurance, can be achieved. Not only that, by integrating with the PAT, we can able to get inline online testing, such as in case of the blending stage, if we put the NIR, then no need for the I mean, sampling, 100% sampling, what we are making over there, subsequently analysis is to be carried out over here. No need for that. This will give the instant result because of the probe by putting into that. And similarly, in granulation stage, this will give the granulation size, or if roll compaction is there, particle size, this precise particle size which is required for that, this will be given instantly. No need for the offline testing in this case. And same way, dissolution by developing this by models, different models, and in the coating as well as the different instant results, it comes through the PAT. Design process optimization, traditional approach. This is one factor at a time. This is end product testing, whereas empirical approach it is. And QBD approach is a systematic approach, whereas this is a multi-factor at a time. Interaction between the variables is being studied in this. Third hybrid approach, this reduces not only cost, but also it reduces the time as well. Take in simple terms, if 60 kg batch size is there, this can be divided into this initial blending stage. This can be adopted by traditional approach because number of variables is only one. Whereas here, drying granulation or this can be adopted by QBD approach. A QBD approach, a combination is this both two. Because over here, number of variables is more and the batch size is the continuous. That's not the concern for that. And this strategy, generic opportunity for in the stability stage, modified Arrhenius equations, humidity corrections. This is associated with isoconvergent paradigm. Isoconvergent paradigm means time to failure to formation of equal amount of degradants in every conditions. Earlier, historical, if you consider this equation was up to this was inversely proportional to the T, and this rate of kinetics is not linear. At higher temperature, it was getting non-linear, and because of that reason, this cannot be extrapolated. Prediction of the self-life cannot be made with the historical. Here, this sensitive humidity correction is there. As a result, it gives the complete linear in nature, which within 2 weeks you can able to estimate the prediction of self-life, rather than waiting for the ICH thumb rule, 6 months wait for the accelerated and 1 year to wait for the long term. This is the protocol for this accelerated modified Arrhenius degradation kinetics. Different temperatures with the combination of RH related to humidity and number of days. This result is to be put into the Monte Carlo simulations, and it will extrapolations to make precise within 2 weeks the results. Self-life determination can be made. This is the one case study. Different strength is considered over here, and as per the ICH, this comes to 0.5 specific impurity limit, qualification limit we can able to give, but isoconvergent limit, it comes with this humidity correction. Arrhenius degradation, it comes to 0.12 averagely it comes to, whereas historical it comes to 0.2. Different packaging material that is considered over here, and all or this degradation level is less than 0.2, except this 2 which is the PVC material. Yeah, this is another study, revolutionary study. LC, this LC to be connected with the QBD platform, automated QBD platform, and the automated data exchange between these two QBD software takes place, which provides the report, and visuals. There's a no transcription error possibility is not there. There's no human interpretation, no human involvement is there. And as a result, validations, doing the validation 40% time saving is takes place, whereas method development over here, it comes to which takes place for the new products 60 days. Within 2 weeks, method development for the new complex product can be carried out. Outsourcing, rapid access of technology, rapid access of the equipments. This is the strategy. At the early stage, if this company is mid-size, small size, and early adoptions for the QBD in the development stage, this is the way, leveraging the prior public knowledge from the big company, and based on the core competency which is available, this externalization and virtualization trends, this is depending on that. Externalization, if some specific test is to be outsourced, whereas the virtualization, this is all test from the R&D that is to be given to other company, outsourced, that is to be outsourced, virtualization, this card. Informatics platform study. This is a cloud-enabled software based. Data can be accessed from anywhere, from the company, anywhere. This is the difference. These days, this has got also outdated LIMS, ERP, manufacturing execution systems. Reason behind that, ELN, there's a trending report customized solution is possible with this, whereas these are the systems where it's software. So time saving can be made, speed and data accessing, decision making, customized solution, it is possible to making use of this. Implementation, supplier risk monitoring, continuous. This is another strategy with generic opportunities, is that comprehensive view of the supplier risk in order to get complete risk analysis, quality, regulatory rated, financial health, industry, business, operational performance, and visibility. This is a visibility of complete risk from the supplier, whereas in traditional it is narrow focused, subject to a reactive process in nature. This is another strategy for that, generic strategy. This is developing an early indicator of product quality. Currently, it is a competitive method for the CFU count, as well as it's a detection and it's a counting. It's a competitive method is 100 years old method, and the time consuming. It takes a 7 days at least, whereas by a rapid non-destructive automatic method, by that within 1 hour detection can be made and counting can be carried out by autofluorescence method. Develop better biomarkers. Current biomarkers is optimization is based on the hypothesis, because it is associated with the diagnostic process. Here it is related with genomic expression, hypothesis of free diagnostic. It saves the time tremendously. It's related with bioinformatics. Implementing a process improvement methodology. Traditional method is focuses on productive period, whereas a continuous, it's a non-productive period, which is the principle of the Lean Six Sigma. Lean, this reduces the wastages, Six Sigma, by reducing the defects and streamlining the process, continuous improvement. That is the way learning organization culture is to be maintained. Developing quality matrices, risk and escalations, whereas quality matrices access at present qualitative or subjective in nature, which is a focus on the QC systems whereas systems and processes, and now it is trend is getting changed for the manufacturing quality matrices. If you consider here, last week only, in 28 July, FDA releases some guidelines, 10 baselines based on some baseline, which is more quantity based and objective based, and focuses on the sites, product, and systems, rather than only it is related with the QC systems or processes. Here it is related with the sites also included into that while developing the matrices. Self-auditing, this is the one step to improve the perception for the regulatory. Yes, August 2014, FDA has proposed a priority review program, considering that volume, considering the complexity of submission, and not only for the resources of the FDA, but also the industry intent also considered. Intent means different criteria. They have considered for that, which is a clear, specific, consistent criteria. Also, in such criteria, priority review is to be given. This is proposed for that. It is still under draft. This manual policy and procedures, and this is given, not yet finalized. If single cycle regulatory review, if it lasts 4 or 5 years, 2009 to 2014, if it is analyzed files and how many times review was made, it is submitted to FDA, has come back, then it averagely it comes to every file, every approval, it is associated with the 5 times. In order to make it single, FDA has initiated some measures, that is real-time communication. This real-time communications means prospective communications, information exchange with the FDA prior to the formal actions. This reduces the number of cycles review. This reduces, it enhances the transparency as well. Here, this 30 days with information request, and this 45 days it gives for the review, 45 days for the review for the. This starts and end. This is for the major as well as the minor variations, minor corrections if it is there. The total days is 255. This time will be the same, but this can be adjusted for the generic if they are proactive in nature. This can be adjusted from before and after as well. Concluding thoughts, because the QBD doesn't address this, there needs to be some measures, expansion of QBD is must, so that they take care not only quality, cost of goods, sustainability of the process, scalability of that. That is possible. QBD is supposed to be extended further to the development by QBD, development by design rather than quality by design. Thank you.